Mendel's work on transmission of traits, paper, cell biology, genetics, molecular biology, and evolution. Mendel's work on transmission of traits. Outcomes: increased understanding and comprehension of the basic principles and concepts of genetics and inheritance. Improved ability to interpret and analyze complex genetic data like Punnett squares. Enhanced critical thinking and problem-solving skills as students learn to apply Mendelian genetics principles to solve problems. Improved understanding of genetic basis of inherited disorders and diseases. Enhanced research skills as students conduct, understand the experiments, and analyze the data related to Mendelian genetic principles. Objectives: to understand the fundamental principles of genetics and inheritance as established by Gregor Mendel, to explore the basic concepts of Mendelian genetics such as alleles, dominant and recessive traits, and genotype-phenotype relationships, to appreciate the relevance of Mendelian genetics in various fields such as medicine, agriculture, and conservation biology, to apply Mendelian genetic principles in problem solving and experimental design, to develop critical thinking skills and scientific reasoning by studying the Mendel's experiments. Gregor John Mendel. Gregor John Mendel, 1822 to 1884, was an Austrian monk who gained posthumous fame as the father of genetics. Mendel conducted hybridization experiments on pea plant in the backyard of church between 1856 and 1863. From these experiments, he established some rules of heredity, which later became known as Mendel's principles of heredity or Mendelian laws of inheritance. Law of segregation, monohybrid ratio. Law of segregation states that during gamete formation, the alleles for each gene segregate from each other so that each gamete carries only one allele for each gene. Mendel crossed tall and dwarf pea plants. These tall and dwarf plants are pure lines. That is, Mendel crossed a homozygous tall capital T plant to a dwarf small t plant. This generation is called P1 or parental generation. The offspring produced on crossing the two pure line parents having contrasting traits, that is, the tall T and dwarf small t represent the F1 or first filial generation. The F1 generation consisted of all hybrid tall plants T. Mendel self-pollinated the F1 generation tall plants to produce F2 or second filial generation. Explanation: hereditary character is controlled by unit factors that exist in pairs in the organisms. A specific factor T T exists for the trait tall and another for the trait dwarf. You can see here capital T gives the tall and small t gives the dwarf individual. These factors, now called genes, occur in pairs. This kind of cross is called monohybrid cross because a single pair of genes or alleles are studied for their inheritance. The trait that is expressed in F1 generation is called dominant trait. Tallness in the above experiment is a dominant trait. That phenomenon is called dominance. The other trait which is not expressed phenotypically is called recessive trait. Dwarfism is a recessive trait. Mendel observed that 1064 F2 plants out of those 787 are tall and 277 were dwarf. The ratio of tall to dwarf is phenotypic, that is phenotypic ratio is 3 is to 1 in the F2 generation. The genotypic ratio is 1 is to 2 is to 1, that is capital T is equal to 1, capital T small t is equal to 2, and small t is equal to 1. During the formation of gametes through meiosis, a pair of alleles, genes, or factors separate or segregate so that each gamete receives only one of these factors. A homozygous dominant plant capital T produces gametes with the dominant factor capital T, and the recessive plant small t produces gametes with the recessive factor. If the individual is heterozygous capital T small t, two different types of gametes are produced in equal proportions. Each gamete contains either capital T or small t. Thus, law of segregation is later also called as law of purity of gametes. You can see here this plant produces capital T small t gametes, and this plant produces again the heterozygous capital T small t. And they cross together, they produce three tall offspring, that is phenotypically tall, capital T, capital T, capital T, small t, and one dwarf offspring, that is small t, small t. The ratio is phenotypic ratio is 3 is to 1, whereas genotypic ratio is different, 1 is to 2 is to 1. In Mendelian inheritance, a monohybrid cross is a genetic cross between an organism that is heterozygous for one trait, capital T small t, and another organism that is homozygous recessive small t for the same trait. The purpose of cross is to determine the genotype of the heterozygous organism. For example, let us say that an organism with the genotype capital T small t exhibits a dominant trait such as tallness. To determine the genotype of this organism, it can be crossed with another organism that is homozygous recessive for the same trait, such as dwarf plant, small t, small t. If the ratio of the tall to dwarf plants in the offspring is 1 is to 1, then the original organism was heterozygous. If the ratio is 0 is to 1, all tall individuals, then the original organism was homozygous dominant, capital T, capital T. If the ratio is 1 is to 0, all dwarf, then the original organism was homozygous recessive. The monohybrid test cross is a useful tool in determining the genotype of an organism and in understanding the inheritance patterns of a single trait. You can see in this diagram the test cross, wherein the offspring is mated with the double recessive parent so that it will give the whether that offspring is homozygous or heterozygous, that individual is homozygous or heterozygous, it can be understood. So this kind of cross is called test cross. Law of independent assortment, dihybridization. Law of independent assortment states that genes or factors for different traits segregate independently during the formation of gametes. Experiment. The cross involving two pairs of contrasting traits is called dihybrid cross. In one of his crosses, Mendel crossed pea plant with yellow round seeds Y, and another with green wrinkled seeds y, small r. All the F1 generation are yellow and round phenotypically and double heterozygous genotypically Y, R. So round is dominant over wrinkled, yellow is dominant over green phenotypes. The F1 individuals are self-pollinated to produce F2 generation. Approximately 9 by 16 of the F2 plants are yellow round, 3 by 16 are yellow wrinkled, 3 by 19 are green round, and 1 by 16 are green wrinkled. Phenotypic ratio of F2 is 9 is to 3 is to 3 is to 1, and the genotypic ratio is 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. Explanation. The F2 offspring shows not only the parental combinations, that is yellow, round and green wrinkled phenotypes, but also new combinations, namely yellow wrinkled and green round phenotypes. These new combinations are produced due to genetic recombination among the factors by independent assortment. The yellow round parent has genotype Y, R, and the green wrinkled parent Y, small r. Since each parent is homozygous for both characters, color and space, each will produce only one type of gametes. The Y, R parent will produce all Y, R type gametes, and the small y, small r will produce all small r type gametes. All F1 hybrids resulting from the fusion of these gametes would be double heterozygous with the Y, R genotype and appear yellow round. Both the dominants Y and R expressed themselves in F1, while both the recessive alleles y and r remain hidden. The F1 dihybrid would produce four types of gametes. You can see here the four types of gametes: Y, Y, small y, R, and small y. This the in equal proportions. When these gametes mate randomly and they produce these offspring. This is the principle of independent assortment of characters. There will be four types of male gametes, four types of female gametes formed by the F1 dihybrid. So these are the four kinds. These are male gametes. These are female gametes. These are female gametes. During self-pollination of the F1 dihybrid to produce F2 generation, as shown in the Punnett checkerboard, this can be grouped into four kinds on the basis of phenotypic appearance, like yellow round. You can see yellow round in these corners, yellow round individuals, and yellow wrinkled. You can find these are the yellow wrinkled. Then again, yellow wrinkled individuals, and green round individuals can be found here. Green round, green round. And again, you can find the green wrinkled here, only one individual. This is also yellow round. So this is how the ratio phenotypically it is 9 is to 3 is to 3 is to 1. 9 yellow round, 3 yellow wrinkled, 3 green round, and 1 green wrinkled individual. Literally, dihybrid cross does imply two monohybrid crosses, the test cross of dihybrid. In the above dihybrid cross, two monohybrid crosses are one for color of the seeds and other for the seed shape of the seed. If these characters assort independently, each character, even in dihybrid cross, does exhibit monohybrid phenotype and genotype ratios in the F2 generation. If the color of the seed is considered, the F2 phenotype ratio is 3:1 and the genotype ratio is 1:2:1. Only shape of the seed is considered, the phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. This proves that law of independent assortment is correct. You can see here the double heterozygous dihybrid, yellow round and green wrinkled individuals. They produce all different combinations in equal proportions, 1:1:1:1. Conclusion. The lesson titled Mendel's work on transmission of traits discussed the pioneering experiments of Gregor Mendel, which established the basic principles of inheritance, which provided a foundation for modern genetics. This lesson details Mendel's experiments, the concepts of dominant and recessive traits, and laws of segregation and independent assortment. The lesson highlights the scope of Mendel's work, including its impact on the field of genetics and the development of genetic engineering and biotechnology. Thank you.